Our next speaker is Dr. Sergei Kulemzin, graduate from NSU in 2006 and received his PhD in molecular genetics in 2014. Since then, he's been exploring the field of cell therapy, collaborating with several leading hospitals in Russia and Eastern Europe countries. He and his colleagues has developed a novel chromic antigen receptor for the therapy of B-ball, NHL, and prostate cancer, and T-all.
So let's all welcome Dr. Kulemzin.
Thank you so much. I'm really excited to share some of our research and also some of my ideas on how structure of chimeric antigen receptor affects efficacy of cell therapy. So I won't really dive deep into the basics because Dr. McClellan shared a lot of info about CAR T cell therapy. I would just like to highlight the core difference between T cell immunity and CAR T cell immunity. So with T cell immunity, when T cell are facing tumor cell, they detect peptides in MHC and then they attack tumor cell. So with CAR T cell, we are using chimeric antigen receptor to induce all these reactions, the reactions of cytotoxicity, the proliferation. So let's take a look at the core elements of chimeric antigen receptors. First of all, we have promoter. That is a sequence of DNA that actually will induce transcription of RNA encoding chimeric antigen receptor. Professor McClellan shared a lot of data about how you can use promoters, how you can combine bidirectional promoters to produce two CARs simultaneously. So it's really quite important to put right promoter into your constructs. Then you have antigen binding domain that will recognize target on tumor cells, transmembrane domain, co-stimulatory and signaling domain. So we have cytotoxicity, proliferation, expression, and specificity, what will be determined by different parts of chimeric antigen receptors. So if we compare situation with normal T cell tumor cell interaction and CAR T cell tumor cell interaction, we see that in the case of normal T cell, a lot of signaling domains are separated. And in case of chimeric antigen receptor, stimulatory domains and co-stimulatory domains are both changed together in one chimeric antigen receptor. So with normal T cell activation after facing tumor cell, we have separated signaling for cytotoxicity proliferation. In case of CAR T cell, all the signaling events are induced by just few parts of chimeric antigen receptor. So what situation is really more beneficial for anti-tumor immunity? Let's take a look. So if we compare different designs of chimeric antigen receptors that were well known and are in established CAR T cell product, we see that three of most popular CAR T cell product that was in well use for like more than 10 years, there is not a lot of really difference. So all of these three chimeric antigen receptors are employed FMC63 as antigen recognition domain. They all use quite similar hinges and transmembranes. The core difference is that Axicell is CARTA are employing CD28 signaling, and two other products are employing 4-1BB signaling. So, it's really interesting what type of chimeric antigen receptor is better, because when we are working with other drugs, we can compare, we can really tell that like ofatumumab will be better than rituximab. In case of chimeric antigen receptor T cell therapy, it's really hard to establish the best drug. And the core problem in comparing different chimeric antigen receptors is that we don't have side-by-side clinical trials. We don't have clinical trials that will compare in completely similar setting different products, different CAR T cells. That's why it's still challenging for us to tell which product is better. When we are performing some meta-analysis, we can see that Yescarta is a bit better. It's also induced more CRS, so it's tougher for patient, but it seems like it's more efficient in controlling tumors. But in different meta-analysis, we see better CR rate for CD28 signaling products for Yescarta. But if we take a look at the persistence of these CAR T cells, it's really intriguing that Yescarta will purge from human body faster than Kymriah. So 4-1BB signaling is more beneficial for long-term effects. So if we are talking about thousands of days of persistence, then CAR T cells based on 4-1BB signaling, it seems to be better for patient than CAR T cells based on CD28 based signaling. Which one is better is still challenging question to answer, because in long-term perspective, it seems like CD28 signaling is better than 4-1BB signaling. But if we look at the further perspective, it could be more beneficial for patient to use milder signaling to ensure that CAR T cells could persist for longer time. But again, there is no side-by-side comparison. That's why, for me, it's what's really interesting when I realized that one Chinese team were able to compare different designs of CAR T cells by combining these CAR T cells and infusing it into the same patient. So they made a real, not even side-by-side comparison, but they made a comparison of similar CAR T cell product manufactured from same T cells and then infused into the same patient. And I think that such layout of design of comparison of CAR T cell therapy could be really beneficial for us, because with such design we can compare efficacy and persistence of CAR T cell therapy on a small amount of patient with some quite intriguing results. So here we see that 4-1BB CAR T cell product are usually have better expansion than CD28 cell product. And the persistence of 4-1BB based product in same patient will be in most cases better than persistence of CD28 product. Unfortunately, with 7 patients that were enrolled in this clinical trial, they were not able to detect any significant changes, any significant difference. So they can't really tell us, please choose one design over our design. But I think that we really have to think about employing similar idea of combining different CAR T cell product and using it with the same patient to ensure that one product is really better in terms of persistence, because we see a lot of papers in Nature every year with some novel insights into CAR T cell design, with some novel ideas of how to make the ultimate CAR. And then we don't see all these great improvements in the clinic, because no one know will it be really more beneficial for a patient or no. And each clinical trial to ensure efficacy of one product is better over efficacy of another product usually costs a lot of money, because you have to enroll several dozens of patients into the age group. With this idea, with combining CAR T cell product and infusing it, combination of CAR T cells into same patient, I think we can obtain a lot of details, a lot of data, even from small phase 1 clinical trials. So how about difference between chimeric antigen receptor signaling and TCR, rec-TCR signaling? There is one really interesting paper when they compare different designs of CAR chimeric antigen receptor specific for CD20, and rec-TCR also specific for CD20. So we can see here that rec-TCR is yellow lines, that efficacy of CAR T cells are higher than efficacy of rec-TCR T cells. So it seems from this data, like obvious that CAR T cell therapy will be more efficient, and CAR-induced signaling is more efficient than TCR-induced signaling. But, surprisingly, it's not really the case. There is another interesting paper when they were able to produce T cells that are expressing both EBV-specific TCR and ROR1 specific CAR. And then they manufacture synthetic particles that were covered with corresponding ligands for chimeric antigen receptor and TCR. And then they were able to titrate efficacy of signaling determined by calcium flux, depending on the density of these antigens. And surprisingly, in contrast to the previous data, you can see that even small density of the ligand for TCR can induce faster and stronger signaling than even higher density of ligand for chimeric antigen receptor. So here we see that TCR-induced signaling could be better for T cell activation because it could be more, it could be natural. And then we see the idea that when we first designed our CAR T cells with CAR of second generation, of third generation, of fourth generation, we were using the approach of Lego. We were just taking different building blocks of different receptors, of different parts of signaling receptor of immune system. Then we were mixing and matching in. But we never really take a deep look on how harmonic, how natural this signaling will arise. So in typical layouts of chimeric antigen receptor, signaling is really only one-sided. We have 3 items in CD3 zeta. We have strong CD28 for signaling, but all this signaling could be really not too beneficial to induce long-term proliferation of T cell. We can induce strong cytotoxicity, but then T cell will be overactivated and will eventually die. There is another interesting paper that were taken a deep look at to which CD3 subunit could be better for CAR T cell design. And we see here that our typical CD3 beta subunit is by all means not the best choice to make a chimeric antigen receptor. It will result not into the best tumor purge compared to other CD3 subunits. What I'm trying to tell is that we used to believe that chimeric antigen receptors T cell therapy, it's well established that we have products that are on the market for almost 10 years, and that we believe that we have really optimized and matured technology, but it's not really the case. The problem is that the major efforts are making towards some well-established chimeric antigen receptor designs, and we have thousands of patients treated with just a few types of chimeric antigen receptor T cell design. We don't have a lot of patients treated in side-by-side comparison clinical trials to check whether some changes in CAR T cell design could be more or less beneficial. So usually we have just clinical trial with, I don't know, 10 to 20 patients treated with novel CAR T cell, but the results from this clinical trial are not really comparable with other clinical trials. And in the end, the investigators, the investor, the governmental regulator can't really tell whether this type of novel therapy will be more beneficial or less. So in the end, they still are using some well-established CAR T cell design. And we are not really evolving because of that. So we still have same design of CAR in clinic that were developed 20 years ago. and a lot of novel and interesting designs that are less tonic, that are that could produce more persistent CAR T-cell efficacy are not in use just because we can't really prove the efficacy. It's too expensive to run clinical trial with 100 patients in one group and 100 patients in another group to prove that your novel CAR is more efficient than any other designs of CAR. So we really have to think how to make some kind of platform for comparing different CARs in one setting so we can move faster in the direction of building a better CAR. I would like also to point your attention that CAR T-cell therapy, in contrast with classical chemotherapy, radiotherapy, and kinase inhibitors, are aligned not into the tumor biology, but just on the surface of tumor cells. And that's why we can really apply novel approach on how to purge tumor cells, not relying on their biology and not being really afraid of tumor cells to become resistant. So we have to employ the idea of CAR T-cell therapy, I think, wider and wider because of that. CAR NK cells are by all means now very hype topic. And one of my colleagues just called CAR NK cells like holy grail of cell therapy, because a lot of teams, a lot of companies and clinics are looking into CAR NK cell therapy. So we were working with NK cells and CAR NK cells for quite a lot. We were trying to build better NK cells to be carrier of CAR. We made one research with NK cell lines, it's YT NK cell line, when we were making knockout for SHP2 gene. And then we compared how CAR NK cell with SHP2 and without SHP2 can eliminate tumor cells. So you can see here, so with the model of NK resistant, the U145 PSMA expressing prostate cancer cells, if you knock out SHP2 from NK cell, you can have much better efficacy of tumor elimination compared even with CAR NK cell, but with SHP2. So the rational design of NK cell biology, I think, really important to make a better and more efficient tools for cell therapy. Also, we are working with novel tools to treat tumors of T-cell origin. You know, it's quite hard to treat with standard protocols of chemo and radiotherapy. So CAR T and CAR NK therapy could become an option for TLL and PTCL treatment, but there is also some challenges. On target of tumor activity eliminates normal T cells, and that could be a really life-threatening condition for patient. But I think that it's fine because in most cases, CAR T-cell therapy is considered a bridge for hematopoietic stem cell transplantation. So if we remove normal T cells along with tumor T cells, it could be fine since patient will soon be transplanted with novel HSCST. Another challenge is that CAR T cells are T cells themselves. So if we are targeting T cell antigens like CD5, CD7, CD3, then we can just purge all CAR T cells as well. That's why NK cells seem to be a good option for therapy of this cell or of tumors of T cell origin. So what we were doing in the area of CAR T cell therapy toward T cell tumors, we made a novel CAR T, novel chimeric antigen receptor specific for CD5. CD5 is a standard marker of the majority of tumors of T cell origin. We employed a murine as if we developed previously by our team for this purpose. We were making this chimeric antigen receptor in collaboration with Professor Filatov, who established murine hybridoma specific for CD5. Then we check efficacy of this chimeric antigen receptor on model NK lines YT. we detected activity of one design of this chimeric antigen receptor, and then we moved to murine studies. We decided to compare current K cells and CAR T cells efficacy in side-by-side experiment because it's also not too much papers comparing CAR T and current K cells in similar settings. So we incorporated IL-15 co-stimulatory protein membrane form of IL-15 into our CAR just for the purpose of better persistent and to make current K cells persist better in model animals. Then we infused model animals with surrogate cells labeled with GFP and luciferase. And in 7 days, we infused model animals with either current K cells or CAR T cells and proceed to visualization. So to our surprise, current K cells were less efficient in eliminating T cell tumors compared to CAR T cells. We see that at some moment, current K cells weren't able to control tumor growth. Then they were controlling tumor growth a bit, but still overall efficacy of CAR T cells was much better compared to current K cells. And it's really quite surprising data for us because we were also believe that NK cells is superior because they are more cytotoxic. They have some intrinsic capacity to detect and kill target tumor cells. But it seems like the difference in proliferation capacity, the difference in persistence would really play a role. So CAR T cell therapy could still be more efficient, at least in some cases, for oncohematological conditions. And again, I love the idea of Professor McClellan on combination of CAR T cells and just normal untransduced NK cells. Or maybe in future, we can just make a product not from apheresis material, not from T cells, but from the mixture of T and NK cells and just use a mixture of current K and CAR T cell for therapy. So persistence of tumor cell in case of CAR T therapy and current K therapy in different tissues was a bit different, not significant difference between persistence of tumors in CAR T and current K treated animals, but still we see some tendency in selected animals. So that's all about current K and CAR T cell comparison. And I believe that we have to think more about design of clinical trials, think more on how we can extract more information and how can we move novel designs of CAR T cells and current K cell in clinic faster. Thank you so much.
Thank you very much, Dr. Sergei, for providing an excellent discussion. Now, I believe there will be some questions from our audience.
Hello, good afternoon, Sergey. Once again, thank you for your talk. So I just have a few questions regarding your presentation. So first, you mentioned that clinical trials may not be capturing the best, what do you call this, like the best actual clinical performance of these different designs of CAR T cells. One thing I was thinking, are there clinical trials out there that are already trying like different combinations of CAR T cells at different time points? Like for example, go with the 4-1BB cells first, and then I don't know, CD28 first, and then once they've already like delivered targeted most of the tumor, and then you go with the 4-1BB so that you have greater persistence. Are there clinical trials out there that have already done this? And also, there was in your presentation a while ago, I just wanted to clarify that group that tested the 4-1BB and the CD28 CAR T cells side by side from the same source. Did they infuse both types into the patient, or they chose one or the other? Yeah. Sorry, you're on mute. Sergey, you're on mute. You're muted right now. We can't hear you. Sorry.
Sorry, I was mute. So regarding second part of the question, in this Chinese publication, they were combining both CAR T cells with CD28 signaling and 4-1BB signaling. They were using combination of CAR T cells of both types in one single infusion in one patient. Regarding your ideas about signaling and about switching from different signaling to another signaling, from I can switch in my points of view from pure biologist, fundamental biologist, to more like clinicist. So from my point of view of biologist, it would be exciting to have like different CAR T cell product for same patient, and then using first maybe one type of CAR T cell product, checking for persistent efficacy CRS, then switching to another type of CAR T cell product. But from my point of view of clinician and biotechnologist, I would say that it's just impossible, because it will be too expensive, to manufacture and perform QC of even one CAR T cell product. It's a huge money. If we want to manufacture different CAR T cell product and switching from one to another, it will be just, incredibly costly to treat one patient. But the idea is really interesting. We were discussing with our colleagues just recently, a few weeks ago, that in future, the idea of solid CAR design could evolve into the idea of library of CAR employed for one patient. So we can imagine that we are making a product that comprised library of chimeric antigen receptors with different signaling domains. And we are manufacturing this CAR T cell product, and we infusing this product into as a single product, performing one QC. We are infusing this product into one patient. So if one subpopulation of CAR T cells with one signaling is better and more efficient in elimination tumor cells over other population, just this population will expand. And in one patient, maybe one population of subpopulation of CAR T cells will expand. In another patient, another subpopulation will expand. I think that we can try to make something of this next step product of non-homogenic CAR T cells. It will be really interesting.
Okay. Yeah, thank you very much, Sergey.
Thank you, Professor, for Dr. Destur. In patients receiving treatment, how significant are cases for CAR T resistance, and what subset of patients are actually, if there were, in this case, what were their conditions that led to the treatment failure?
It's really interesting, and it's interest for multiple researchers on how to select patient for CAR T cell therapy, how to know which type of therapy will be more beneficial for patient, one therapy over other therapy, how to choose CAR T cell therapy. So we know that at least for CD19 specific CAR T cell, there are two major reasons for CAR T cell therapy failure. One reason is that tumor cells become escaper cells, so they become CD19 negative. And that was the case for bispecific CAR T cells that were presented today by Professor McClellan. So in this case, we can help to treat patient with escaper cells by either switching to another type of CAR T cell product with another specificity or by employing bispecific CAR T cells. Another major reason for CAR T cell resistant in patient is that T cells are not good enough, are not fit enough, because we are using autologous CAR T cells. We are making CAR T cell product from patient's own T cells, and in some cases, patients are not not fit enough. The patient's T cell after multiple lines of chemotherapy could be not too active in terms of proliferation. So after we infuse these cells into the patient, proliferation rate could be suboptimal, and persistence of CAR T cells could be also suboptimal. So it could be a really good idea, and we were discussing with regulators in Russia and in Malaysia that it could be really interesting to cryopreserve T cells, or at least PBMC, for the majority of patients with leukemias and lymphomas before chemotherapy. I mean, we can try to cryopreserve PBMC for all patients that are diagnosed for T cell or B cell lymphomas, leukemias, and we can cryopreserve material PBMC from this patient before proceeding to chemotherapy, because in multiple cases we see that patients are waiting and struggling to be treated with CAR T cell therapy, but technologists can't manufacture products. We know that in some clinical trials, 20 or 25% is a normal rate of failure to manufacture CAR T cell product. So with this approach of switching to cryopreservation of PBMC for all patients before proceeding to chemotherapy, I think we can partially solve this second reason for CAR T cell resistant.
Are there any more questions from the audience? Thank you very much, Dr. Kulemzin, for accommodating the questions from our community. To show appreciation of your time and effort to be one of our special guests this afternoon, we would like to present you with a certificate of appreciation.
Certificate of Appreciation for Dr. Sergei in recognition for your insightful presentation on clinical insights of CAR T cell therapy, presented at the 6th floor Agustin Barcelon Auditorium, the Medical City, Ortigas, November 21. We are grateful for your values contribution to the success for this event. Signed by Dr. Destura. Thank you very much, Dr. Sergei.
Thank you very much. Thank you very much. It was a great pleasure for me to be here.
So guys,